Welcome to Sciencera. In this video, we're going to discuss the citric acid cycle. The citric acid cycle. Citric acid cycle, Krebs cycle, or tricarboxylic acid TCA cycle is the most important cyclic metabolic pathway for the energy supply to the body. It is the universal pathway for aerobic metabolism as the cyclic series of the reaction. Citric acid cycle essentially involves the oxidation of acetyl coenzyme A to carbon dioxide and water. The enzymes of TCA cycle are in the mitochondrial matrix. Basically, it involves the combination of 2-carbon acetyl coenzyme A with a 4-carbon oxaloacetate to produce a 3-carbon tricarboxylic acid citrate, which is followed by the oxidation of 2-carbon to carbon dioxide and regeneration of oxaloacetate. Let's look at the reaction of TCA cycle. The citric acid cycle proper consists of a total of 8 successive reaction steps, each of which is catalyzed by an enzyme. To begin with, all the organic fuels molecule is converted to acetyl coenzyme A. In case of carbohydrate metabolism, pyruvate so produced from glycolysis undergo oxidative decarboxylation to form acetyl coenzyme A in presence of multi-enzyme complex pyruvate dehydrogenase complex. This step is connecting link between glycolysis and the citric acid cycle. If you are new to my channel, welcome to Science Era. I post videos twice a week. You can also join my Telegram group with the link given on the screen, where you can find free notes, textbooks, past paper, and also can contact me personally. The enzyme catalyzed reactions. The cycle starts with the condensation of a carbon 4 oxaloacetate and carbon 2 acetyl coenzyme A. Oxaloacetate reacts with acetyl coenzyme A plus water to yield a 6-carbon citrate plus coenzyme A in presence of a regulatory enzyme, citrate synthase. Citrate is isomerized into isocitrate through the intermediary formation of the tricarboxylic acid, cis-aconitate. The isomerization takes place in two stages: dehydration of citrate to cis-aconitate by aconitase, which remain bound to the enzyme, and rehydration of cis-aconitate to isocitrate by the same enzyme, aconitase. Then isocitrate is oxidatively decarboxylated to a carbon 5 compound, alpha ketoglutarate, through the intermediary formation of tricarboxylic keto acid, oxalosuccinate. This is the first of the 4 oxidation reduction reaction in TCA cycle. The reaction takes place in 2 stages: dehydrogenation of isocitrate to oxalosuccinate, which remains bound to the enzyme. In this stage, NAD plus and NADP plus is required as electron acceptor. Decarboxylation of oxalosuccinate to alpha ketoglutarate is the second stage. Both the reactions are irreversible and are catalyzed by the same enzyme, isocitrate dehydrogenase. In the next step, alpha ketoglutarate is oxidatively decarboxylated in a manner of analogous to oxidative decarboxylation of pyruvate to form carbon 4 thiol ester, succinyl coenzyme A, and carbon dioxide by enzyme alpha ketoglutarate dehydrogenase complex, or alpha KDC complex, which is in the mitochondrial space. The step is physiologically irreversible step of the cycle. One of the peculiarities of the citric acid cycle is that it contains two successive oxidative decarboxylation step, which is step number 3 and step number 4. Succinyl coenzyme A undergoes an energy conserving reaction in which the cleavage of its thioester bond is accompanied by the phosphorylation of guanosine diphosphate or GDP to guanosine triphosphate GTP. The reaction is catalyzed by succinyl coenzyme A synthase, which is succinic thiokinase. The enzyme involves the formation of an intermediate, succinyl phosphate. The phosphate is transferred first onto the imidazole side chain of histidine residue in the enzyme and then onto the GDP producing GTP. The generation of a high energy phosphate from succinyl coenzyme A is an example of a substrate level phosphorylation. In fact, this is the one reaction in citric acid cycle that directly yields a high energy phosphate. Then succinate is dehydrogenated to fumarate by flavoprotein enzyme. Succinate dehydrogenase located on the inner mitochondrial membrane. The enzyme contains the reducible prosthetic group flavin adenine dinucleotide or FAD as the coenzyme. FAD function as the hydrogen acceptor in this reaction. Fumarate is hydrated to form L-malate in the presence of fumarate hydratase, formerly known as fumarase. Fumarate hydratase is highly specific and catalyzes trans-addition and removal of hydrogen and hydroxide. This is the fourth oxidation reduction reaction in the citric acid cycle. The other three reactions being step number 3, 4 and 6. Here L-malate is dehydrogenated to oxaloacetate in the presence of L-malate dehydrogenase, which is present in mitochondrial matrix. NAD+, which remain linked to the enzyme molecule, act as a hydrogen acceptor. Net reaction of TCA cycle can be written as given on the screen. Energy yields per pyruvate molecule. Energy yield in step number 3, where isocitrate is converted to alpha ketoglutarate and carbon dioxide. Method of ATP formation involved the respiratory chain oxidation of NADH. ATP yield per molecule in step number 3 are 3. Step number 4, where alpha ketoglutarate convert into the succinyl coenzyme A and carbon dioxide by respiratory chain oxidation of NADH. ATP yield in this step is also 3. Step number 5 of the cycle, succinyl coenzyme A ADP plus phosphate gives you succinate and ATP. Oxidation at the substrate level, ATP yield per mole is 1. Step number 6, succinate is converted to fumarate, a respiratory chain oxidation of FADH2. Energy or ATP yield per mole is 2. And last step is step number 8, malate is converted to oxaloacetate, respiratory chain oxidation of NADH, ATP yield is 3. So 3, 3, 1, 2, 3 gives you total gain of ATP equals 12. Regulation of TCA cycle. Several factors serve to control the rate of reaction sequence in citric acid cycle. These are described as number 1, substrate level. One of the controlling feature of any reaction sequence is availability of the various substrates involved in it. The relatively restricted concentration of oxaloacetate puts an emphasis on its role in controlling the input of acetyl coenzyme A into the cycle. Regulation of the rate of this reaction would control activity in the enzyme cycle. Then on the enzyme level, All mitochondria form from widely different sources, possess constant relative proportions of various enzymes, including the characteristic dehydrogenase of the citric acid cycle. The observation suggests that there exists a genetic mechanism for the control of synthesis or integration of the key mitochondrial enzyme during mitochondriogenesis. The genetic mechanism may involve a single operon containing all necessary structural gene to control the enzyme biosynthesis. Then there is respiratory control. Respiration rate depends not only on the nature and concentration of the substrates to be oxidized but also on coupling of respiration to phosphorylation. Intact mitochondria are usually tightly coupled so that their rate of respiration is controlled by the ratio of ADP over ATP. When this ratio is high, respiration is promoted. In contrast, low ratios where high ATP concentration is there, it declines the respiration. Added ATP can even inhibit respiration because they bring out reverse electron flow. These phenomena are now known as respiratory control of the TCA cycle. Accessibility of cyclic intermediates. The activity of citric acid cycle is also controlled by its accessibility to acetyl coenzyme A of intermediates of the cycle. The mitochondrial membrane itself provides a mean for the admission of some substrate at the exclusion of others. A few examples are given. Like mitochondrial succinate dehydrogenase is freely available to succinate from outside the mitochondria but not to fumarate. Furthermore, added fumarate is also not freely accessible to mitochondrial fumarase. Ketosis. The accumulation of ketone bodies, acetoacetate and acetone, formed by the liver in diabetic results from the production of more acetyl coenzyme A than can be cyclized via the Krebs cycle or other synthetic reactions. Under these conditions, the rate of Krebs cycles slow down, probably due to the hormonal action since ketone body formation. For example, ketosis is affected by hormones by hypophysis and adrenal cortex. Control of enzyme activity. Three enzymes, namely citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase, regulate citric acid cycle. Citrate synthase is inhibited by ATP, NADH, acetyl coenzyme A, and succinyl coenzyme A. Isocitrate dehydrogenase is activated by ADP and inhibited by ATP and NADH. Alpha ketoglutarate dehydrogenase is inhibited by succinyl coenzyme A and NADH. Moving on to the features of TCA cycle. TCA cycle is an open cyclic process. TCA cycle is a cyclic process, however, it should not be viewed as a closed circle since many compounds enter the cycle and each intermediate of the cycle connecting another metabolic pathway. Being the open cyclic process, there is no compulsion of acetyl coenzyme A to start the cycle. The cycle can initiate from any other intermediate. Amphibolic nature of the citric acid cycle. Citric acid cycle provide various intermediates for synthesis of many compounds needed by the body. Krebs cycle is both catabolic and anabolic in nature, hence regarded as amphibolic. TCA cycle is actively involved in gluconeogenesis, transamination, and oxaloacetate and alpha ketoglutarate respectively serve as the precursor of this for the synthesis of aspartate and glutamate, which in turn are required for the synthesis of other non-essential amino acids like purines and pyrimidines. Succinyl coenzyme A is used for the synthesis of porphyrins and heme. Mitochondrial citrate is transported to cytosol where it is cleaved to provide acetyl coenzyme A for biosynthesis of the fatty acids, sterols, etc. Requirement of oxygen in citric acid cycle. There is no direct participation of oxygen in the cycle. However, the cycle operates only under the anaerobic condition. NAD plus and FAD from NADH and FADH respectively required for the operation of cycle can be regenerated in respiratory chain or electron transport chain only in the presence of oxygen. Therefore, citric acid cycle is strictly aerobic in contrast to glycolysis, which operates in both aerobic and anaerobic conditions. Role of vitamins in TCA cycle. 5B vitamins are associated with TCA cycle essentially for yielding energy. Number one is riboflavin in the form of flavin adenine dinucleotide FAD, a cofactor for succinate dehydrogenase enzyme. Niacin in form of nicotinamide adenine dinucleotide NAD, the electron acceptor for isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and malate dehydrogenase. Thiamine as thiamine diphosphate required as a coenzyme for decarboxylation in the alpha ketoglutarate dehydrogenase reaction. Lipoic acid, it is required as coenzyme for alpha ketoglutarate dehydrogenase reaction. And lastly, pentothenic pantothenic acid as part of coenzyme A, the cofactor attached to active carboxylic acid residue such as acetyl coenzyme A and succinyl coenzyme A. This brings us to the end of citric acid cycle. Thank you for watching. If you like my video, don't forget to subscribe for more.